There we go. All right, so if you've taken a biology course before, you know, you might have had an introduction to the plasma membrane. I'm going to tell you something. You're going to have a whole new appreciation, or maybe it might bring feelings of dread when you're learning about the plasma membrane. But the plasma membrane is more than just an envelope that surrounds the cell and keeps everything on the inside. All right? It's a limiting barrier. Okay? It prevents things from moving in and out of the cell. And there's a reason for that. All right? So we're going to learn about that this semester. But before we get into that, we need to understand the makeup. All right? What makes up our plasma membrane? Okay? And so when we were talking about chapter 2 about fats or lipids, we learned about the different types of lipids. All right, we talked about phospholipids, for example, all right, which is a huge component of your plasma membrane. So in fact, your plasma membrane is made up of, okay, equal parts by weight of lipids, fats, and proteins. Because remember, I'm not going to pull up the slide here, but if you can recall all the different functions that proteins have in our body. Enzymes, so they're great for chemical reactions. All right, transport, because we have channels in our plasma membranes. So certain things can enter and exit the cell. And that's one of the reasons why. So strictly by weight, where is it? By weight, all right, we have equal parts of fat or lipids and proteins. Okay? So this is a huge concept that I really want to hammer home to you, this next point. All right, it regulates movement of not all substances, but most substances, okay, that can move across the plasma membrane or move in and out of the cell. That's crucial, because some things don't need, all right, to be regulated to get across the cell, meaning if something's really, really small, okay, and if it's what we call nonpolar, it can move across the plasma membrane with no problem, right? It doesn't need help or it doesn't need permission. Okay, the plasma membrane selectivity will not affect that. Okay, so that leads me to this point here. All right, if you are really, really small and nonpolar, okay, then you can move across the plasma membrane without any help whatsoever. Okay, nonpolar and really small. Okay, so when we talk about the lipid component of our plasma membranes, here are the big ones. The phospholipids, those are the most numerous. Okay, cholesterol, that's good. We're going to talk about the role of cholesterol. It helps to, believe it or not, all right, add integrity and strengthen your plasma membrane, especially during extreme temperature changes. Okay, and then glycolipids. Glyco meaning carbohydrate. It's a lipid with a sugar group attached to it, a carbohydrate group attached to it. Okay, so those are the different types of lipids that are present in our plasma membrane. All right. So when we talk about the phospholipids, and again, it's nicknamed here the balloon with two tails, so we kind of draw it, again, circular with these two wavy tails that are coming off of it. All right? So the polar part of a phospholipid, all right, is going to be hydrophilic, water-loving. Okay? Hydrophilic, water-loving, which is very good to see because water is in high abundance on the outside of the cell and inside the cell. So the polar heads or the hydrophilic heads will make contact with water. All right, and then our tails here, those are nonpolar. Okay, and those are hydrophobic. We talked about that in chapter 2 when we were talking about water being the universal solvent. All right, and so water, well, going back to polarity, right, non-polar substances are going to be water-fearing. They don't like water. Okay, so if we have water in high abundance outside the cell and inside the cell, all right, I'm going to come right back to this slide here. If we look at our plasma membrane, it's got this type of configuration to it. All right, we call it the phospholipid bilayer. All right, here are our phospholipids, all these purple balls with the tails hanging off. I'll even zoom in more. Okay. All right. And they have this configuration, this whole thing right here, that where I'm drawing that square. Okay. Inside the square, that's the plasma membrane. And so you have one layer of phospholipids, all right, and the heads are facing out of the cell. And we have another layer of phospholipids on the inside of the cell, and the heads are facing inside the cell. So the water-loving portions, okay, are going to be in contact, because like I said, there's a lot of water outside and a lot of water inside. Okay? So those hydrophilic heads are going to make contact with the water areas there. Okay? But the internal portion of our phospholipid, all right, membrane here, okay, that is going to be the hydrophilic portion. All right, the tails there, that's the water fearing. Okay? So let me go back to the slide here. Okay? So keep in mind, all right, we call it the phospholipid bilayer, bi meaning two, so there's two layers of these phospholipids, all right, and they are positioned, the phospholipids, tail to tail, heads on the outside of those tails there, okay? So in those tails, we do have an internal environment of the membrane. We really won't talk about what goes on in there too much, so you're not going to have to really worry about that, okay? But our polar heads are going to be directed outward, all right, and that's going to be what's going to come in contact with our water. All right, so a couple terms that you really need to understand. This one right here in bold print, interstitial fluid, all right, that is the name of the fluid that is outside of the cells. Okay, so any tissue material outside the cell in that area, all right, is also referred to as interstitial tissue. Okay. And then on the inside of our cell, that's where our cytoplasm is, all right? That fluid, all right, environment is the cytosol. Remember, the cytosol is that viscous-like fluid that is one of the three components of the cytoplasm, okay? So the cytoplasm is the overall term that we use to describe the internal environment of the cell. All right, and there's three components to it. Cytosol being the liquidy part, okay? Organelles, all right, those are all those little structures that we talked about, the Golgi apparatus. Remember, we broke it down into membrane bound and non-membrane bound, and I went through smooth endoplasmic reticulum, rough endo, all that stuff. Those are the organelles. And then the third component are what we call inclusions. And those are kind of like the leftovers that are just floating around. It could be random amino acids, it could be ions, they could be, I was going to say glycerin, not glycerin. Glycogen, the storage form of glucose, is an inclusion. Okay, and it just floats around there in the cytoplasm. Okay, all right. So the other two components of our plasma membrane, all right, cholesterol, all right. Again, its role is to help to strengthen that membrane. Okay, so when extreme temperatures, either really hot environments or really cold environments, and this is why I tell folks all the time. All right, when I was younger, I was always told cholesterol is bad. Well, there's good cholesterol and bad cholesterol, but I'm telling you this right now, all right, cholesterol is good, way better than sugar, okay, way better than sugar, because you need cholesterol. You just saw one of its purposes. Cholesterol is needed for plasma membrane, all right, but it also, we need it to make certain hormones, okay, very important, our steroid hormones. So me being the only male here, I'm going to make testosterone, I need cholesterol. For you ladies, if you're going to make, all right, estrogen and progesterone, you need cholesterol, all right? It is a precursor, something that you need to make these hormones, all right? Bile salts, all right, which are, what do I want to say? Bile salts are a digestive component that you need when you eat a fatty meal. Your liver makes the bile, all right, and then it gets stored in your gallbladder. But when you have a fatty meal, all right, it acts as what we call an emulsifying agent. It helps to break down fats. Right? And bile salts, when we make those bile salts, right, we need cholesterol. So my point is cholesterol is important. It's not as bad as you think, okay, in moderation. Right? And then finally, the glycolipids, all right, we have a carbohydrate group attached onto our lipid there, all right, and it helps to form what we call the glycocalyx, which is, and this is only going to be on the outer phospholipid layer. It's not on the inside of the cell, it's always on the outside of the cell. And the glycocalyx, another name for it is the sugary coating to our cell. Okay, helps to identify the cell. All right, make sure that the cell is a self, S-E-L-F cell, all right, and not a foreign cell. Okay, so our body doesn't kill it off. We need to be able to identify the cells that belong and which ones don't. All right, glycocalyx helps with that sugary coating on the outside of your cell. All right, questions so far? Anybody at home with questions? Hopefully you're paying attention to me. Not watching Jeopardy. Well, Jeopardy starts at 7:30. Okay. So I'm jumping in this picture here, what we saw just a few moments ago, and this is pretty much showing you the plasma membrane. Whoops. All right. So again, you have your phospholipid layer, all right, the bilayer on both sides, and then you have, here's one of our glycolipids, all right, only on the outside of the cell. And then these big blue structures that you see that are floating around, those are your proteins. All right, we're going to talk about those in a moment. And then you can see kind of like these little ball-like structures that look like they're hanging off of a string. All right, that's your cholesterol right there. Again, it helps to strengthen and add integrity to our plasma membrane, which is really important. Okay. All right. So, question: what portion of phospholipid molecules form the internal environment of the membrane? All right, that would be the nonpolar hydrophobic tails. Okay, nonpolar hydrophobic tails. All right, so what are the two characteristics that something needs to have in order for it to move across the plasma membrane without any help? One of them is listed up there on the board. Yeah, what's another thing? Small, that's right. Has to be both of those, small and nonpolar, and it can get through the plasma membrane. Won't need any help. Okay, because we're going to talk about, all right, when something is polar or if it's large, then we're going to talk about how it gets on the inside of the plasma membrane. Okay, so the first part of the plasma membrane is going to be the lipids. Now we're going to talk about the proteins. Okay, this makes up the other half of the weight of the plasma membrane. Okay, and they float within the plasma membrane or on top of the plasma membrane, much like trash does on water. Okay, I forgot the name of that big trash pile out in the Pacific Ocean. They actually named it. It's so big. All right, but it floats on top of the Pacific Ocean, and that's pretty much how our plasma membranes do. All right, so we're going to see, all right, a lot of the functions for the plasma membrane are going to involve these proteins. All right, I'm guaranteeing you that after this semester, you're going to have a huge appreciation for proteins. All right, if you're ever taking a test and it's asking you about something and you have no idea what the answer is, and proteins listed in one of the answer choices, might as well choose it. All right, might as well. Okay, because proteins do a lot. Okay, so there's two types. All right, we've got the integral, and that means that it's going to span the entire width of our plasma membrane. And then we have the peripheral, which means it's on the periphery. It's either on the inside of the plasma membrane or it's on the outside. Okay, so if we go back to this slide here and we see the pictures, all right, here's an integral plasma protein. This guy spans the entire length. Here's another one. All right, and then here's a peripheral. See how it's floating around on top on just one part? And then here's another peripheral one down in here. Okay? Easy so far, right? Okay? So a reminder, remember, these proteins are going to have specific functions for the cell and the plasma membrane. Okay? So the integral proteins, all right, these guys are embedded within the whole entire cell. Okay, they span the width of it. All right, and so therefore, parts of these proteins are going to have hydrophobic and hydrophilic regions. All right, it should say hydrophobic. It says hydrophobic and hydrophobic. It should say hydrophobic and hydrophilic up there. Okay. All right. I'm sorry, I'm jumping ahead here. Here's the hydrophilic. So the hydrophobic region of the protein is going to interact with the hydrophobic region of the plasma phospholipid bilayer there. Okay. So several of these types of proteins are what we call glycoproteins. They have a carbohydrate group attached to it. Right? And so those carbohydrate groups attached to these proteins, right, will have specific functions. One is a cell identifier. So we know, all right, for example, blood typing, ABO, blood typing, okay? That's going to be one of our functions, okay? So that sugar group helps to identify. And when our white blood cells are constantly floating through, all right, our body, and they're moving in and out of tissue, and they're floating through the circulatory system. All right, their job is to check on all these cells. So remember when I said that you have about 37 trillion cells in your body? All right, so imagine all your immune system has to monitor, all right, those cells to make sure that they're supposed to be there. Okay, so they're going to go around and check. You know, they're basically like the secret police. You know, do you have your papers? Identification papers, please. We need to identify you. And so they go around and they just identify if the cell is supposed to be there. All right, and you have these glycoproteins on the surface of the cell identifying what that type of cell is. And those white blood cells will look at that and say, okay, you're supposed to be here. I won't kill you this time. All right. Then we have our peripheral proteins. All right. They're not embedded throughout the whole bilayer. They will either be on the internal or the external surface. One, all right, or the other. All right. It's either in or it's out. Real simple. Okay. All right. So we're going to go and now talk about, all right, some of the different types of categories for these proteins. All right. So for the next little while, I'm going to be teaching you folks, all right, a couple of important concepts here. Okay. So pinch yourself, make sure you're wide awake for this because we're going to really get into some really important information here. Okay. So the first functional category is going to be transport, which we're going to be talking about a lot in this semester. And guess what? You're going to talk a lot about it in 211 too. 211 also. Okay? So we're going to learn when we have something that's too big, or if it is polar, how can we move it across the plasma membrane? Not to worry. Transport proteins will do that for us. They're going to move substances right across that plasma membrane there. All right, and we're going to learn about all these different types, the channels, the carriers, and the pumps. Symporters, which I just got done explaining to one of my 211 classes, and I apologize to them because I'm only going to briefly talk about it here, but it comes back, all right? I don't want to say to haunt you, but you'll see that the symporter again later on when you're learning about how thyroid hormone gets made, okay? It's really important, and there's actually a genetic trait, all right? I don't remember which gene it's carried on for that specific pump. It's really cool. All right, cell surface receptors, okay? Think of a lock and a key, okay? Think of the cell surface receptor as the lock, all right? And then the key is going to be what we call a ligand, which are these special types of molecules that will essentially unlock that receptor there, okay, and cause something to happen. Maybe it's going to release something, or maybe it's going to inhibit something being made, okay? So we see that when we talk about the neurotransmitters. Right? When we talk about an action potential, which is an electrical signal that travels down the cell, and then it gets converted into a chemical, all right, signal. Okay, so we're going to learn about that. All right, identity markers, that's what I was talking about before. All right, this is how, all right, our cells know, all right, the white blood cells specifically, if, all right, our body cells, or we call somatic cells, are supposed to be where they're in our body. Okay, so it's also another way for us to identify healthy cells from unhealthy cells. All right, cancer cells all the time. Cancer cells used to think, oh, you know, I'm cancer. I can divide really fast, uncontrollable division, and I just grow and grow and grow. And then the immune system's like, nope, no problem. I can find where you are cancer because you're not expressing the proper identity markers, so now you're dead. So cancer cells have gotten clever now because now what will happen is, and some of these viruses are doing the same thing, that when they take over a cell, they start to pop out, all right, the normal identity markers. So when the white blood cells come rolling through checking things, they're like, oh, all right, you look normal, but in secret, it's not, all right? So it's pretty important that you have, all right, these identity markers because otherwise you're dead. All right, enzymes, another function of proteins that we will talk about, all right, quite a bit. And the important aspect of enzymes is this right here: catalyze chemical reactions. Okay, which means they start. It's like starting a fire. That's what a lighter is. A lighter helps you to start a bonfire. All right, these enzymes will do the same thing. Okay, So they'll be either on the internal portion or the external portion of the cell, and in some cases, they're going to be inactivated. So they're not going to do anything. But then another protein comes along and activates it, turns them on, and then we can start some chemical reactions in the body, okay? So enzymes are really important. Anchoring sites, remember, cells need to have, remember we were talking about tissues, cuboidal and columnar cells, how cuboidal cells are cube-shaped, okay? Columnar cells are like taller than they are wide. There's a reason for that. Those cells have a cytoskeleton, much like we have a skeleton, all right? But those tubules, the microtubules, okay, that cytoskeleton, those intermediate filaments and all they have to attach to something, okay? Otherwise they'll just float around, all right? So they have anchoring sites on the inside of the plasma membrane. So the cytoskeleton can attach to that, okay? And then, especially when we're talking about epithelium, one of the characteristics of epithelium, and I know we haven't covered this yet, is that epithelium is tightly packed. If you looked at over any of your tissue slides, okay, you'll notice, all right, when we were going over like simple squamous, doesn't matter if it's simple or stratified, but all those cells were tightly packed to one another, okay? And if they weren't attached to one another, they'd be slipping and sliding and falling out of place. There would be no organization. So the cells have to attach themselves to one another on the sides. And that's where the cell adhesion proteins come into play. All right, it's like a seamstress. They sew, all right, the cells together so they don't move. And so nothing, because I'm going to tell you this right now, okay? You've got bacteria, and as we know, viruses that are on the outside that want to get in, okay? Now, we have certain mechanisms that help to prevent that, okay? But sometimes they get clever, and they can find a way in. And if they find a way in, all right, I won't say they'll never come out, all right, but it can cause problems for us. And one way is if they can try to get in between the cells, they'll do it. So if you get swelling, inflammation, okay, that will push the cells apart, and that's a problem. One of the ways to prevent that are the cell adhesion proteins there. They keep the cells tightly packed. So here you can see some of the plasma membrane proteins, plasma membrane proteins. I'll zoom in. You can see now we're going to put in picture form some of their functions. Okay, here you can see, all right, channel, carrier protein, pump. These are your transport proteins. And so in this example, you're seeing stuff that's outside the cell moving in. Okay, protein carrier, this is perfect for glucose. See this right here? That's the little symbol that we use for glucose. It's got 6 carbons, it's a hexose ring. Okay, so these carrier proteins help for us to get glucose from outside the cell inside the cell. That's hugely and largely important. Glucose is the preferred energy source for cells. And your brain loves glucose. It gets top billing. It's the #1, all right, recipient of glucose. And in fact, other organs in your body and tissues will forego, all right, taking glucose in just so the brain can get glucose. It's called the glucose sparing effect. All right, really important. All right, and then you have a pump. All right, and the pump moves things in and it'll move things out of the cell. So those are some examples of transport proteins. Okay, here you can see a cell surface receptor. This is our lock and key. All right, so here's the lock, the blue protein here, and the ligand is a key. It could be some sort of chemical, some sort of molecule, anything. All right, but when that happens, it can start a chain reaction. It can turn something on, it can turn something off. All right, it can cause the cell to get rid of stuff. It can cause the cell to take stuff in. Okay. Our identity marker, all right, you have these specific carbohydrate groups attached to the protein, allowing us to identify what the cell is. And then we have an enzyme, okay? So this protein will act like an enzyme. The substrate, which is the starting thing, all right, like metal is to a key, all right? You take, a clump of metal, and then you do something to it, and you wind up with a key. All right, the clump of metal is our substrate. Our product is the key. That's what happens here. All right, anchoring site for our cytoskeleton. You can see here it's on the inside of the plasma membrane, and then you have all these different cytoskeleton proteins attaching to it, giving that cell shape, structure, integrity. And then finally, the cell adhesion protein. All right, attaches one cell to another cell. Okay, keeps things from getting in between the cells. Because usually on the other side of the cell layer, you're going to have the bloodstream, because remember, right, capillary, well, we haven't talked about capillaries, but certain blood vessels are going to be located throughout the whole body, and if whatever that pathogen, if it can get between the cells and get into the bloodstream, then it can go anywhere in the body, and that can be bad. All right, so what type of plasma membrane protein provides the means for moving materials across the plasma membrane? Those are transport proteins. Okay? Remember, if you're not sure and protein is an answer choice, pick it. Okay, let's take a deep breath. I'm going to take a sip of water while you're doing that, because we are going to move into, all right, membrane transport. All right, membrane transport is going to be crucial for your understanding when we get into nerves and muscles. Okay, so I'm really going to try to hammer this home with you folks. All right, so if you don't understand it 100%, that's okay. Okay, but I'm going to do my best. All right, and if not, all right, don't worry. Watch the video. Okay, and that will help. Even the review video. All right, so going back to what I said initially when we were talking about the plasma membrane, all right, keep in mind it is a selective physical barrier. Okay, that means it'll let certain things in and out of the cell and prevent others from entering in and out of the cell. Okay, and it's a physical barrier between the external cell environment, what we know as the interstitial fluid or space. There's also another name for it, okay? The extracellular space, okay? And then the internal environment, where the cytoplasm is, the cytosol, all right? Or the intracellular space, meaning within, okay? A couple terms there, all right? So again, it regulates what comes in and out. Now, when we talk about certain types of cells, and especially when we get into the nervous system, we're going to talk about this next term here, the electrochemical gradient. If you break that word down, electro and chemical, that means we're going to be dealing with literally electricity, all right, and chemicals. All right, I know that seems very simplistic, but it'll make sense when we get into chapter 12, okay? So it also is huge for cell communication. And there's a concept of what we call secondary messenger, all right? And I'm not going to talk about this now. You'll learn more about it in chapter 17 in 211, all right? But that cell communication, right, it's going to allow for certain chemical processes to occur, to have the cell undergo certain functions, okay? It'll also allow for signals or nerve signals to move across the cell. More on that, though, in later chapters. So let's talk about membrane transport. All right. So we have to get stuff to come into the cell, and we have to get rid of stuff. What would a cell be eliminating? Name me something that a cell would want to get rid of. Or what's your body want to get rid of after you've eaten meals? Waste. Yeah, you want to get rid of, and that's what cells, same thing. And also, some cells will make things. Like, perfect example, a beta cell in the pancreas makes insulin. So it's going to want to eliminate the insulin. It's going to want to, pump it out into the bloodstream, especially after you have a pizza. I was talking to one of my sections the other night. When I was growing up, we had 4 food groups. Okay, it was meats, fruits and vegetables, dairy, and bread. Right, but now we have the food pyramid. Okay, now I think they use a plate. I think that's the newest one. Point being is, when I was taking a health class, my instructor told me that pizza had all 4 food groups on it. So that's why I use it. Right, pizza will help to release insulin. Okay, it causes a release of insulin because there's proteins, mainly because there's sugar, glucose, carbohydrates. All right, but we need to have that, all right, when that occurs, the beta cells sense that, and then they're going to pump out insulin so the rest of the cells in your body can now start to gobble up or take in glucose if their glucose levels are low. Well, we use glucose to make energy, ATP, all right? So same thing, insulin helps. Think of insulin as putting stuff into cells. lowers blood sugar levels, okay? So the two types of categories that we're going to talk about are passive and active processes, okay? Passive, and I'm going to go through the characteristics of each because it's really important that you know that. I'm going to start off with the general characteristics. We're going to start off with passive first. Now, wow, you look at this picture here and it's overwhelming. But I want you to make a note that figure 4-7, start with figure 4-7 when you are studying this material, okay? And I say this because when we talk about membrane transport, how we get stuff across the plasma membrane, all right, this flow chart kind of explains just some of the basics, the basic material, all right? So we start off with passive processes and active processes, all right? And it will tell you some of the characteristics. For example, passive processes do not require energy. That's good, all right? And they move substances down their concentration gradient, which means if I have 100 things in one hand and one of that same thing in my other hand, I have a high concentration gradient of the stuff in this one hand and a low concentration gradient in the other, okay? You have more of something, all right, somewhere, and you have less of that same thing in another space, okay? It's like the front seat and the back seat of your car, all right? You could have 10 people riding in the back, 1 person in the front. You have a higher concentration gradient of humans in the back seat and a low concentration gradient of humans in the front seat, okay? So that's what we're talking about when we talk about the concentration gradient. I'm not going to go through this slide, all right, but just go through this, try to learn it, memorize it if you need to, all right? And then, because I'm going to go through this all right now, okay? But when you're on your own, if you have a good understanding of this flow chart here, okay, then you'll be good. You'll be good. Because I'm telling you, a lot of this material can be intimidating. All right, so let's talk about some of the characteristics of passive processes and active processes. Okay? Passive, again, does not require energy. Does not. And then you're going to move something, all right, down that concentration gradient. All right? So an area where you have more of whatever the substance is, glucose, for example, sodium, right? For example, we're going to revisit this concept, banana floating in the ocean. Remember that. When we get to the nervous system, I'm going to talk a lot about that. But you have a lot of sodium outside your cell, and you have very little of it inside the cell, normally. Okay? So because you have a lot of sodium outside the cell and very little inside the cell, nature always wants to be in balance. Okay? Wants everything to be fair. Okay? If you have $100, well, let's make it easy. If you have $99 and I have $1, wouldn't it be fair if you gave me, all right, $44, all right? Oh, actually, what am I saying? $49, all right? Because then we'd have, you know, the same amounts. Wouldn't that be fair? All right, so that we would be equal. So that's what happens here with our concentration gradient. All right, we want it to be the same. So ideally, sodium wants to have 50% on the outside of the cell and 50% on the inside of the cell. So it's going to want to move into the cell because there's more outside than there is inside. So it wants equilibrium, balance. All right. So the two types of processes that we're going to talk about is diffusion and osmosis. When you see the word osmosis, you're going to think of this right here. Water. Okay? If there's a test question, all right, and in that test question it mentions nothing about water, and one of your answer choices is osmosis, you better not pick it. All right? Osmosis involves water. It has to be mentioned. Okay? Okay. So the active processes do require energy. Okay? And they are going to move a substance up its concentration gradient. Easy way to remember that? Think about pushing a boulder up a hill. Okay? That's going to take energy. Right? Does it really take a lot of energy from you if once you push a boulder down the hill, once it rolls, you're done, right? It doesn't take any energy. All right, but if you have to roll that boulder up the hill, that's an active process. That requires energy. Okay? So think of it like that. Substance is going to be moved up its concentration gradient, so therefore it requires energy to do so. Okay? When we are going down our concentration gradient, when you start to roll, all right, that ball down the hill, once it starts to roll on its own, you're done. It does not require energy. Okay? So we're going to talk a little bit about what we call membrane-bound vesicles, or vesicular transport, which means something on the inside of your cell is going to make something, it's going to package it up, and then it's going to send it away. Now, it might send it to the plasma membrane because maybe it was damaged and we need to repair it, or maybe we need to send whatever that cell made outside the cell because it's a hormone and it has to go somewhere. All right? So vesicular transport, anything that involves, all right, vesicular transport, vesicles, packages, involves energy. All right? That's an active process. And we're going to talk about that too. Good so far? Do I need to repeat anything? Am I going too slow? Okay. All right. I have a tendency, I've been told in my reviews, and by the way, I do read my reviews, every single one of them, at the end of the semester. And I do my best. Oh, by the way, I keep saying by the way. I was thinking, because I asked you folks last class, you want to just stay here instead of moving. I think it's a little bit quieter here. It just makes it easier. You don't have to go upstairs. All right. I know there's not a lot of room in some of these desks, but it just makes it easier, especially if you're like me and I get easily distracted. If there's noises and voices. I hear the voices all the time. All right, let's talk about diffusion, the first of our passive processes. Okay? So diffusion, all right, is going to involve a concentration gradient. So that means we have a lot of one substance in one area, and then very little of that substance in another area. So since we're talking about cells, let's just use that. Inside the cell and outside the cell, okay? So outside the cell, we have a lot of something, and inside the cell, we have very little of it, okay? So what will happen with diffusion is cell, whatever that substance is, it's going to want to move from that area of higher concentration to lower concentration. So I'll go back to sodium. Okay? A lot of sodium outside the cell, very little sodium inside the cell. So where is sodium going to want to go? Is it going to want to move out of the cell or move into the cell? It wants to go down its concentration gradient. Okay? So what do you think? Well, it's going to want to move down. Okay? Think of it like this. Here's our concentration gradient. You have 100 molecules of sodium, actually 140. I'll use simple math. And 1 here. Okay? So it wants to move down its concentration gradient. So I'll draw our cell here. So you have about 140 micromoles outside the cell and about 20 inside. Okay? So can we all agree there's more sodium outside the cell? And there's less inside. Perfect. So now we have a concentration gradient. Okay? Now if we had, all right, what is that? 160 altogether. So if we had 80 micromoles of sodium outside the cell and 80 micromoles of sodium inside the cell, all right, it would be equal, right? You'd have the same amount inside the cell and outside the cell, so there would be no concentration gradient. There'd be no movement of anything, okay? Because it'd be equal. Doesn't want to go anywhere. But now we look here and we see there's more sodium outside and less inside. So where does sodium want to go? Inside, that's right. So that's just another way of saying sodium is going to move down its concentration gradient. It's going to go from an area of high concentration here to an area of low concentration here. You guys like that? Is that good so far? Okay. So now sodium is what? Do you guys remember what sodium is? It's an element, all right? All right. It's a molecule, all right? Actually, specifically, it's an ion. Okay, sodium, the chemical symbol for sodium is Na. And actually, in our bodies, it's going to be Na+, which means it's a cation. It's positively charged. It has one less electron, meaning it has one more proton. Okay? So it's positively charged. Point being is, when we have concentration gradients and molecules are moving back and forth across the plasma membrane, because they're always trying to go from that area of higher concentration to lower concentration. Okay? When we have these molecules in motion, all right, we call that kinetic energy. Okay? Think of it like this. Going back to the boulder. If I have, drawing this on the computer. So here's my hill again. Okay? And I got my boulder at the top of the hill. It's not moving. In physics, we call that potential energy. Okay? It's not moving, but it has the potential to move. Now we push the boulder, it starts to roll down the hill, okay? Now it's in motion, we call that kinetic energy. Okay, a little physics lesson, real quick, okay? So once the object's in motion, it has kinetic energy. Good, you guys are sharp, I love it. Okay. Hold on, let me catch back up here. Questions so far while I'm scrolling down? Is that good? Do you kind of have a little bit of an understanding? Whoops. Where am I? Here? Yeah. Okay. So, as long as we don't do anything to interfere with the concentration gradient, once those molecules are moving down the concentration gradient, okay? All right, if we don't do anything to interfere with that, we're going to reach that point of equilibrium. All right, and that point of equilibrium, going back to our original example with sodium and the numbers that we were using, once we get 80 micromoles on the inside and 80 micromoles on, 80 micromoles on the outside and 80 micromoles on the inside, all right, it's equal. And when that happens, we'll have no more movement. And that's what nature wants. That's what, all right, sodium wants to do. It wants to reach equilibrium, which means these molecules are evenly distributed on the inside of the cell and outside. Good so far? All right? And I got a picture to help with that. I'm going to show you. All right, it's in your textbook. That's this, right there. So here's a person with a beaker of water. They got food coloring in it, okay? And they put that food coloring initially into the water. Now, would you say there's more food coloring in this area compared to this area? So therefore, we have a concentration gradient. All right, we have more food coloring in that little spot in the center initially. All right, now as time goes on, all right, the molecules for that food coloring will go from an area of high concentration to an area of low concentration. And then eventually it'll spread itself out and be equally distributed throughout the water over time. It takes a little bit of time. All right, but that's what it wants to do. Okay, that's what our molecules want. Okay, so two things are going to affect how fast this happens, the rate of diffusion, we call it. All right, the first one is the steepness of the concentration gradient. So I'll show you an example. All right, here's a real steep concentration gradient. We got something that you have, let's say, 80 and 20, using the same numbers now. Now we have a much less steep concentration gradient in which you have 60 and 40. Okay, it's still they still both total 100, but you can see, all right, you have 60 molecules of something here and only 40 here, so there's not much of a difference. So the rate or how fast it's going to diffuse won't be as fast as it would be with the difference of 80 and 20. Okay, that's 60. All right, the difference is 60, whereas the difference between the 60 and the 40 example is only 20. Okay, so that's what we're talking about. It measures the difference in concentration. Okay, so the steeper the gradient, right, this little ramp that I showed you, the faster diffusion will occur. Okay. The other one is temperature. All right. You ever thrown a cat on a fire that's with hot coals? How fast does it take for it to get out of there? It gets out. It's a bad example, but I used it before in class, and it's better when people aren't wearing masks because the expressions are priceless. But when, all right, think of it like that. All right. When you turn the temperature up, all right. Again, this is kind of going back to physics and chemistry. All right. I know I told you folks that I'm not going to get back into chemistry except for chapter 2. But when you heat up something, all right, you provide more energy to whatever that environment is, because the molecules start to move around faster. And so that's what's going to happen here, all right? So when we increase the temperature, all right, we're going to increase the kinetic energy, okay? And that's going to increase the movement of whatever that environment is. Right? And then, is that a question? Yeah. An example of the body for when you increase the perfect example is when you are exercising. Your body temperature starts to increase, right? Initially, a lot of the chemical reactions in your body start to occur at a faster rate because the enzymes will start to increase. Another perfect example would be glucose, all right? So when your body temperature increases because you're exercising, right, your cells will start to put in these glucose channels into the plasma membrane, and glucose will diffuse from outside of the cell into the cell much faster that way. Yeah. So keep in mind, as we increase the temperature, we increase the rate of diffusion. All right, because the molecules get excited. This is a little bit of an easier concept if you've taken chemistry and physics before. All right, but once you increase the heat, that temperature to that environment, it excites the molecules, increases their kinetic energy. And kinetic energy is that energy that we get from motion. Okay. All right. So let's now talk about a specific type of diffusion. Okay. There's two types. So the first one is simple diffusion, and that's pretty much what I just explained to you. Everything that we were talking about. Okay. So same type of thing, right? When we talk about simple diffusion, obviously it's a passive process, so it requires no energy. But it does, and it also involves the concentration gradient. Okay, so the movement is going to depend on the concentration gradient. Right? So in this case, going back to the cell, right, this is how we're going to move molecules across the plasma membrane between the phospholipid molecules. Okay? So that means if we're going to move something through the plasma membrane, it has to have those two characteristics that are listed up there on the board. They have to be small, and they have to be nonpolar. Okay? If they're small and nonpolar, they go right through the membrane. Okay? So, perfect example is oxygen and carbon dioxide, respiratory gases. Oxygen we need, all right, for energy production, amongst other things. Carbon dioxide is like the waste product. Okay? It's one of the byproducts that we get when we metabolize certain nutrients in our body. Okay? So think of oxygen as the food that you're putting in the mouth, and the carbon dioxide as the other stuff that's coming out somewhere else. Okay? Again, we have fatty acids. Fatty acids are nice because they can move right through the plasma membrane, and that's important because we use fatty acids for a lot of chemical building processes, all right, and also for ATP production. Right? Sometimes we don't like this, ethanol, all right, especially if we've been drinking, all right, but that's why alcohol doesn't need a carrier molecule, it moves right through. Urea is an actual waste product. It's a nitrogen-based product, okay, and so it's a form of cell waste. All right, so simple diffusion, important concept, is not regulated by the plasma membrane. So you can almost assume, I say, that the plasma membrane has no influence on simple diffusion because these substances, I should say, are so small and they're nonpolar, they just go right through. It's like me walking through that wall, okay, if I could, all right, you'd go right through. Okay, so we have this, again, when we talk about diffusion, all right, it will only occur, all right, as long as there's a difference, all right, in the concentration gradient, all right? Once that gradient ceases to exist, we've reached equilibrium, so there won't be any more diffusion, okay? All right, so that's what this picture is showing you. I love this picture here because it shows our oxygen and our carbon dioxide, all right? So when the cells are undergoing metabolism, they need oxygen for their energy processes, so the oxygen comes from outside the cell, it moves across the plasma membrane, and it goes to the inside of the cell. And the cell does whatever it's doing, makes stuff, all right, it's exercising or whatever. And then it has waste products or byproducts from the chemical reaction. That would be carbon dioxide. So you have a high amount of carbon dioxide on the inside of the cell because it's metabolizing stuff and breaking stuff down. And so, and you have very little of it outside the cell, so it's going to go from inside the cell to outside the cell. And it just moves right through the plasma membrane. Boom. Okay? So that's simple diffusion. Plasma membrane will not regulate it. Okay? So that's the first type of diffusion. The second type of diffusion is facilitated diffusion. Okay, facilitated diffusion. All right, now remember what I said before. All right, what about the other things that are outside of our cells? Okay, what about if something's big or if it's polar? We still want to get that inside the cell. So here comes facilitated diffusion. Okay, so that is going to apply when we're dealing with polar solutes and charged molecules or solutes. Okay, or if they're big. I mean, if they're too big, then that could be a problem too. There's another way to get something that's big, like really big, into the cell. Okay, so when we're dealing with small charged solutes or polar solutes, all right, we need help. And who do we go to? Plasma membrane proteins. All right, when you need help, when a friend asks for help, you help them. All right, so in this situation, the plasma membrane proteins will do that. And so they'll be involved in the two types of facilitated diffusion, which is called channel-mediated and carrier-mediated. Okay? Channel-mediated and carrier-mediated. And actually, we saw pictures of those before. When we were talking about the proteins briefly. Okay. Right here. Here's an example of a channel. This right here. Okay? It's like a tube that we stuck right in the plasma membrane and stuff can move through that tube. Okay, that's a channel-mediated protein. Channel-mediated. Yeah. All right. Oops, wrong way. Okay. So let's start off with what the different types of channel-mediated diffusion are. Okay? So, when we are dealing with certain ions, because remember, an ion is a charged particle. Okay, so it cannot effectively undergo simple diffusion. Okay, it needs to be nonpolar and small. All right, in this situation, all right, it's a small ion. All right, it's charged. So we need a channel that is specific for that small ion. Okay, and these channels will be filled with water because remember, there's water inside the cell and there's water outside the cell. You're not gonna be able to stop water. I learned this from a good friend of mine who is a general contractor, and I learned it the hard way when I wanted to have work done on the chimney of my house, and there was a lot of water damage. And even though we took precautions on trying to prevent the water damage, he told me that water goes where it wants to go. Okay, eventually it'll get there. I mean, you've seen the Grand Canyon, all right, what it did there, and that was over a long period of time. Okay, so, all right. When we're dealing with channel-mediated diffusion, you need to understand this right here. Channels are specific to one type of ion, which means, for example, sodium. Okay, potassium can't go through a sodium channel because it's not made for sodium, potassium. It's made for sodium. Okay, so keep that in mind. You have calcium channels, chloride channels. All right, so you cannot get another type of ion to go through a sodium channel. Chloride cannot go through sodium channel. Calcium cannot go through a chloride channel. Okay? It has to be, all right, its own channel that's made specific. Okay? All right. So the first type of channel is what we call a leak channel. Okay? And a leak channel is always open, like 7-Eleven, 24/7, always open. All right, the other type of channel is a gated channel. So if you know anything about a gate could be one of two things. It's open or it's closed. That's it. All right, we call that a binary situation, like a light switch. It's either flipped on or it's flipped off, one or the other. Okay? So in most cases, it'll be closed. The cell's not doing anything and it's supposed to be resting, the gate will be closed. Now, when we need the cell to do something, we're going to open up the gate. Okay? And in order to do that, we need a stimulus of some sort. Could be a chemical stimulus. Okay? All right. So this, we will talk in detail about leak channels and gated channels much more when we get to chapter 12 when we talk about nerve cells and chapter 10 when we talk about muscles. But for right now, I just want you to know this information here. Okay? It'll be exhausting if I were to go through all that information. All right. The other type of facilitated diffusion is carrier-mediated diffusion. Okay? So again, we're going to deal with a small polar molecule, okay? Because it can't go across the plasma membrane on its own because it's polar. Okay? So therefore, it needs a little bit of help, so it finds what we call a carrier protein to help it out. All right? And so what will happen is, I'll use glucose as an example. Glucose, all right, will bind onto this carrier protein, and it'll change shape. All right? It changes its configuration. It's called a configuration state change. Okay? And so glucose binds to it on the outside of the cell, and it undergoes this change, this shape change. All right? And as it does that, it moves the glucose to the inside of the cell. All right? This picture is pretty simplistic. It's a little more complicated than this. All right? But here's your glucose. Okay? Binds onto, all right, our protein carrier here. All right? And it undergoes, see how it's changing shape? It starts off like as a funnel here, and then it closes down, and then the funnel appears on the other side. It's very simplistic, the drawing here, but it's a little bit more complicated than that. But basically, that's what happens. Okay? That's how we undergo, all right, that carrier-mediated diffusion there. Okay? So again, it will, keep in mind, we're dealing with diffusion. So we're always going to be dealing with stuff moving down their concentration gradient. So if you have a lot of glucose on the inside of the cell and very little on the outside, glucose isn't going to enter the cell anymore. Okay? It won't do it. All right? So we have this guy right here, the uniporter. And what that means is uni meaning one. It's going to transport only one substance. Similar to what we saw with our leak channels and our gated channels. They're only going to allow one ion to move through them. Well, in our carrier-mediated diffusion, these carrier proteins are only going to allow the movement of or transport of one substance. Okay? Now, this next concept comes into big, I won't expect you to, you'll appreciate this more in 211, all right? But the number of channels and carriers are going to determine the max rate of substance transport. Which means if the demands, for example, going back to glucose, if the cell's glucose demands increase, if they go up, that cell will put more carrier proteins into the plasma membrane. But eventually it'll max out. It won't be able to meet the demands. And that's what we're talking about here. Okay? So the number, all right, of these channels, all right, or these carriers, it'll eventually max out. All right, therefore, the maximum rate of the diffusion will eventually max out. So if we were looking at it on a graph here, oh, come on, why is it? Oh, please, Lord. It crashed. Son of a gun. Can you guys at home hear me? I know. I don't know, and it's been happening. Oh, maybe they can hear me. Well, I wouldn't know if you could hear me because I can't get on the chat screen. Okay. If you folks can hear me, I am going to stop this really quick. I've got to close the program down. I'll be right back if you can hear me. Give me 1 second. Hey, you guys, are you there? Can you hear me? Okay, cool. Listen, we're gonna take a break just because I had too much. Let's take a 10-minute break. I'll see you back at 7:30. All right. I hate to do that to you guys, but everybody just walked out of the classroom here on me for no reason. So no, I'm just kidding. Let's take a break, and then I'll figure out what I'm going to do about the remaining portion. Okay. So I'll meet you up in lab. Come back and meet me in the lab section. Oh, this is still recording.